in 1997 and received a PhD in physiology from the University of Columbia in 2001. She was a postdoctoral scholar at Brandeis University from 2002 to 2008. In 2008, she joined the faculty of the Department of Neurobiology and Behavior at Stony Brook University and became professor in 2020. She directs the Laboratory for Neural Circuits and Plasticity, and since 2020, she is the Director of Graduate Program in Neuroscience. She is Chief Editor for Frontier in Cellular Neurophysiology and Associate Editor for the Journal of Neuroscience and eNeuro, and is a member of the editorial board of iScience. She is a member of the Society for Neuroscience and the Association for Chemoreception Sciences. Welcome.
So thank you everyone for coming, and I'm really excited to be here with you in person. It's my first talk in person in a really long time. Thank you for being okay with me speaking with the mascot. And so today I'm going to tell you the most recent work that we've been doing in the lab. It started around 4 years ago, and now we're getting ready to put a paper together, and I wanted to show you this work because I think it's a broad interest for a lot of people interested in stuff. So we're going to talk about the taste system and talk about neuromechanisms for taste learning. So why the taste system? The taste system is very interesting to me, and I know to a variety of scientists, because what it does is it transforms something like a small chemical molecule into the detection of the chemical molecule, but also something that can be pleasurable or aversive. It can help you develop food preferences. So something that you ate when you were very, very young becomes your comfort food when you're older. How all of these processes come together is really unknown. So we know how the detection happens, how we detect taste molecules in our mouth. We know some of the circuits that are involved in identifying the taste and telling us what it means to us. But then things get a little bit murky when it comes to what does taste experience do to the brain? Does it influence the brain in any way? Do we learn from taste experience? So let me tell you just a few things about taste and how we detect taste. And then we move on into a more neurobiology, neuroscience type research that we do in the lab. The colors are a little bit crazy because there is a strange interaction between my computer and the monitor. So I have all black in front, and you have these beautiful psychedelic images here. So let's go with that. All right, so we all know that the first thing we do when we're trying to taste something is we put it into our mouth. And we put it into our mouth where we have an organ that is specialized in detecting, which is the tongue, that contains all these lovely papillae. They have different shapes. For example, you see here, there's these circumvallate papillae, which have very sharp grooves, and then a flat part. Then we have this foliate, which have multiple grooves that are coming to the side, and then the fungiform papilla, which is something that comes above the surface of the tongue and has these little coves where the taste receptor cells are located. So you can see them, they're positioned in different places on the tongue. We actually have taste receptors all along the palate, in the back of the mouth, in the throat. We actually have them in the stomach. So they're really distributed across the entire digestive system. We have them in the gums too. So in these papillae, we have taste buds where the taste receptor cells are sitting. So the taste buds are these little budding things that look like an onion like that. They're sitting under the surface. There's a pore. The chemical enters in that pore and activates these cells that look like the membrane of an onion. So these cells do not have an axon. They are just sitting there, and they communicate with the axons of neurons coming from the cranial nerves right there in the end. So what do we know about it? We know that we have a taste receptor cell for each taste. They're very specialized. So we have a taste receptor cell for sucrose. Well, we have, for sweet, a taste receptor cell for salt, one for the taste of umami, which is the taste of protein, one for the acidic tastes, right? They're all conglomerated under the papilla, and they're all ready to receive the taste that when it comes in. Now, they're all distributed in the tongue, and I'm pretty much sure that every one of you in grade school has seen this map that shows how tastes are perceived on the tongue. And so what I'm going to tell you right away is that this map is wrong. We do not have areas that are specialized for sweet and the sides for sour and the back for bitter. What we actually have is that taste receptor cells are distributed across the tongue, and this is really not coming out very well with this conversion in color, because what we really have is gradients of distributions of receptors. So bitter receptors seem to be more heavily distributed in the back, but they're all across the surface of the tongue. The same is true for sour, the same is true for sweet, and the same is true for salty. So the entire surface of the tongue has receptors for all the taste buds and brings that information into the system. So what happens after the tongue, right? Well, what we do have is that our tongue is the place where we have the papilla and the taste receptors. Now, information from here travels into the brain through the cranial nerves, which are those lines. They actually terminate in the brainstem, where there are two nuclei that process information from the cranial nerves. From the nuclei in the brainstem, information about taste comes into the taste thalamus, the gustatory thalamus, which is called ventroposteromedial parvicellular part nucleus of the thalamus. For us, for our friends, is VPMPC. And then from the taste thalamus, the information is coming into the gustatory cortex. It's a part of neocortex that is where neurons respond to taste. In addition to that, we have a splitting of sensory information of the taste system into the amygdala. I don't know if you are familiar with what the amygdala does, but if you're not, I'm going to tell you. The amygdala is the center where we process emotions. So positive or negative emotion related to a certain stimulus get processed in the amygdala. The amygdala and the gustatory cortex are actually really tightly interconnected. So when a taste comes into our mouth, we send that information into the brain. Some of this information also goes to the amygdala, and the conjunction of this information that is coming through the thalamus pathway and the amygdala pathways in the gustatory cortex tells us whether a taste is sugar, whether it's sweet, whether it's pleasant, or if it is bitter, it's quinine, it's bitter, is aversive. We don't really want to have it in our mouth, right? So these are some of the ways in which taste information is processed. So we have talked about the 4 basic taste buds, and again, so this is lemons, okay? So this is citric acid, this is sugar, this is quinine, this is salt, umami, which is taste of proteins, and quinine, which is bitter, okay? But in addition to these basic taste buds, our mouth can also encode information about temperature. You know, some tastes taste different if your beverage is cold or warm, right? Spiciness. Cold, menthol has a cooling feeling in our mouth. We have a taste for astringency, the sensation of astringency from tea, red wine, fat. We have receptors for texture, carbonation, and crunchiness. The chips they're supposed to be. Okay? So all of this information comes from the mouth and is processed into the taste cortex, which is the region of the brain that I study. The important thing about that connection with the amygdala and with other brain areas that tell us about what the taste means to us is that it is important for us to define what is good for us and bad for us, right? If you eat spoiled food, it can potentially make you sick. If you eat something very, very bitter, it could potentially be poisonous, right? If you eat something rotten, it shouldn't be tasting very good, so that you will avoid it. Whereas the pleasant tastings that have all the nutrients that you need to survive tend to have a pleasant taste. So what we distinguish is palatable versus aversive taste. This is what the taste cortex is able to do by being connected to the circuit that we talked about before. Now, one of the questions that my lab is interested in is this one. Does experience with food influence taste preference? We know that experience with something that makes you sick influences your experience. That's another line of research we have in the lab, which studies how the positive nature of a taste can turn negative if it is associated with a stomachache, right? We've all experienced at some point in our life some sort of food poisoning. And you know that if you have had food poisoning to a new taste, the next time you meet that taste, you want to avoid it, right? Those foods, you're never going to touch it. I actually have it for chocolate, but that's because of my brothers. So one of the things that we do not know much about is whether the early experience with food will influence our preferences for food as we grow up, as we become adults. So this is one of the questions we set up to answer in the lab. And we had a few caveats, right? We're not a human subjects lab. We study animal models. So we needed to transfer all of these big picture ideas that I showed you about early experience with taste and preferences in adulthood into something that we actually can examine in the lab. So what will we do? We use mice as a model for studying the taste system. Why are mice a good model? Well, hopefully this picture, this is not coming out very well, but what this picture shows is that the reactions to food of mice, monkeys, and human babies are pretty much the same. If you encounter something that is pleasant, like sweet, the mouse licks its lips and smacks its lips too. The monkey does the same, and the baby does the same. If all these animals and humans encounter an unpleasant taste, they all open their mouth to try to let it out. So the mouse does it, the monkey does it, the baby does it. So there's very conserved reactions to the pleasantness or the aversiveness of food. The other thing is that food is needed to survive for all of these species. All of these species tend to have sweet tooth. Okay, so we are in good agreement across the board. We can use the mouse. The other thing that we need to do is figure out how to translate food tastings into a lab-controlled situation. And so we decided to go for using 3 basic tastings and a mixture to add variety to our potential experience paradigm. So we use sucrose water, we use salt water, and we use citric acid added to the water bottle of the mice at a concentration that is pleasant. So the concentration of oranges, not the one of lemons. And then we use the nutritional milkshake Ensure because this contains calories like sugar, but it also contains fats, proteins, minerals, all sorts of other nutrients that can be needed. So the idea was that with these 4, we have variety that sort of encompasses the entire food pattern. Then what we need is a way to test preference, okay? And it has to be a way that is objective, because the mouse is not going to tell us whether it likes something. or not, or it has its way to tell us through behavior. So what we use is a Davis-Rig gustometer, which is basically a box that has a little slit that can be opened or closed by a moving cord. And it has infrared beams that break when the mouse puts the face inside, and there's a bottle in the back, and we can measure the leaking frequency and the leaking number and all of those parameters about the leaking behavior. In the back of the box, there's this tray that carries multiple bottles. What we do is we fill them with one with water and the other with different concentration of sugar water. And every 10 seconds, the port closes, the rack of the bottles moves to a randomized other concentration that is in the bag. The port opens, the animal can lick for 10 seconds, and then it closes again, we move the bottle, and so on and so forth. And now I don't think the video is really gonna play, because unfortunately you can't really see the mouse, but the tiny little green thing there is the mouse that is licking in the port, and then the rack of the bottles changes, the door opens again, the mouse knows it, goes in and licks for the next solution, and we do it multiple times. And then what we get is a curve, right, that we can measure. Actually, this is 4 overlapping curves, but I'll tell you the reason why it's 4 in a minute. So this is the ratio of the leaking to the sugar tastant versus water for the different concentrations, 0 being water and all the other being concentrations of sugar. And the dotted line is the leaking to water, so the normalization to water. Okay, so as long as the dots are aligned with a dotted line, it means that there's no real difference in the preference for that sugar solution versus water. So the low concentration of sugar, you see there are, the mouse goes and licks them, but it doesn't really prefer them. And then you see that it grows much higher as the solution becomes sweeter. So what this curve is telling us is that the mouse really likes the sugared water much more than the water alone. Okay? And in this case, what we wanted to know was figure out if over the course of development of the mouse, over the ages, so this P is postnatal day, okay, so this is 35, 56, 70, and 91, there's a change in their preference for sugar. And as you can see, there's really no change. So we know that developmentally, without experience, these are all mice that have been reared on their regular food and water. They're all pretty similar in their preference for sugar. They all like it better than water when the concentration is high. So this is the conclusion, and this is our base. So now we wanted to see the effect of taste experience. So here's what we did. We have the animal being born in the lab, and then by postnatal day 21, they are weaned from eating from the mom. They become independent eaters. So now we can change their bottle and give them taste tests, and they will approach it. So what we do is we expose them to the 4 taste tests that I showed you before for 2 cycles. So for 1 day, we take out their water bottle and their food and substitute it with sugar water. The next day, we substitute it with water that contains citric acid. And then the next day to ensure, and then the next day to the salt solution. We do that for practically 8 days, so twice for each taste test. And then we put them back into their regular food and water diet, just like all the other mice. So, and then what we ask is what happens to that superscript that I showed you before, the one that shows the preference over water, in animals that have been exposed to this type of variety type experience with taste tests only for 8 days, only at the time in which they were moving to eat independently. And lo and behold, what we found is that their super preference curve changes a lot. We didn't really expect it to change this much. So the lower curve is the same one that I showed you before before the age of 56, and the one on the top is the one from animals that have been exposed only for 8 days to the variety of tastes. So taste experience really had a profound effect on their preference for sweet. It increases the threshold so they detect sweet much earlier at lower concentration, and they can eat a lot more of it. They prefer it a lot higher. So we did a series of experiments to figure out what is really behind this effect. Is it calories? Right, so we had substituted in our tasting paradigm sugar with saccharin, which is sweet but no calories, and ensure with a comparable concentration of MSG, which is the taste of protein but is not, it doesn't have the same number of calories. I'm not going to show you the data because things get complicated, but I can tell you that no calories doesn't cause the shift. So you need the calories. The other thing you need is variety. We did a series of experiments in which we did 8 days of sugar, 8 days of salt, or 8 days of citric acid, or 8 days of Ensure. So 8 days of sugar alone didn't do anything, 8 days of salt alone didn't do anything, citric acid too, but Ensure, that is by itself calories and variety, shifted the curve just as much as we saw. So for this experience-dependent change in sucrose preference, what we need is both variety and calories. So this is just summarized in the slide, which I put in to give you conclusions and just to slow myself down if I'm speaking too fast. So calories are needed for the shift to occur. A calorie-free option with this early exposure does not show any changes in sucrose preference. Variety is also needed. If we use just one tasting, is not sufficient. So then the next question we ask is, okay, we see the behavior, we have the shift in behavior. What happens to the brain? So to begin to ask that question, we use an approach to measure activity in the animal that is now tasting the sucrose, either without the experience or with the experience. So here's what we do. The animal is positioned under a 2-photon microscope in a head restrained position. So he's just sitting there, the head is still, and there's a spout that comes forward. The mouse does 5 dry licks, so it's just licks at the spout without nothing coming out of it. And at the 5th lick, they get the taster, which is one of the concentration of sucrose. We use only 4 for this because the animal is otherwise gets tired. And we measure the licking raster in the back. So each dot that you see in that plot is a lick. You see that before the blue set of licks, there's 5 licks, which are the dry licks. Then the reward comes and they lick afterwards. So they just lick up the drop of sugar. These animals are not just sitting there. They have had treatment before. One of the treatments is that we inject a viral construct in the gustatory cortex, which sits here on the side, that carries the construct for a calcium indicator. So a calcium indicator is basically a molecule that becomes fluorescent when it detects calcium, so that we can see a calcium signal through the microscope. Okay, the larger the change in calcium, the larger the signal. Big entries of calcium are associated with a neuron firing an action potential, which means it becomes active. In addition to that, they have a prism implanted on the side of the head. This is a microprism, it's really tiny, and what we need it for is, we can't really bend the microscope, we can't really bend the head of the animal, so what we do is we bend the beam of the laser light. So by putting the prism here, we hit the wall of the prism with the laser light that is coming through the 2-photon microscope. This bends, and so it allows us to see the surface of the brain right here. Okay, so this way we can image something that is on the side of the head. The other thing we do is that our mice are transgenic mice. So the white dots are supposed to be green and the rest black. So they, in specific subset of cell, which is the parvalbumin expressing GABA cell, it's a type of inhibitory cell, they express RFP, the red fluorescent protein, so that we can see GABA cells and distinguish them from the ones that are not red, that are all the other cell types, in particularly, for the most part, excitatory cells. So now we can see two groups of cells within the same imaging. All of them are expressing the calcium indicator, and when we're doing the experiment, you see things that light up, okay? So the red cells will stay red, we will see them with the red channel, and the calcium indicator is green, is gonna keep popping up and show us the cells that are being activated when the mouse is licking the taste. So this way we can record and track hundreds, if not thousands of neurons. Actually, the data I'm going to show you in the next picture come from samples of 5,000 neurons in naive animals and 5,000 neurons in animals that underwent the taste experience paradigm that I showed you before. So we have very large sampling of the entire taste area, and we can make calculations about what has happened to the neuron activity when the taste of sucrose comes into their mouth in groups of animals that actually were reared in the regular food and water, and groups of animals that had those 8 days of experience. So here's a quick summary of the data. So what we see is that the inhibitory cells, the red cells, kind of remain the same, the active cells remain the same, but the pyramidal cells, the excitatory ones, become fewer of them are activated by sugar than in the naive group. So that we have a net decrease in the activation of the network following the exposure. This is not all bad, it's actually pretty good, because this activation is done in a way that the system is much better at decoding the concentration of sucrose that is receiving in the mouth. And this is what that plot tells you. So this is a decoding analysis. What we do is we use a set of data, train an algorithm to detect the sugar, to know what activity represents sugar, and then train this algorithm on the remaining data set to figure out if it's better or worse at detecting the concentration of sucrose that is being delivered to them. And what you see in the last plot is the max decoding, which is the peak of those curves. And you can see that the exposure group is much, much better at decoding the concentration of sucrose that is detected. So yes, the network is less active, but it's better at doing its job. So that experience has really changed the functional responses of the neurons. Now, coming down, we want to know how this happens, because this is just, behavior. This is telling us the network, but what is going on at the level of the single neurons and the synapses in the system following that exposure? So what we ask now is, within the neural circuit, what happened? Why is it that we see lower activity? So for this, I have to give you a little bit of background on what cortical circuits look like. And this is a diagram, little schematic that a graduate student in my lab did for a review paper of the taste cortex and its organization. So cortical circuits are organized in layers that are anatomically distinct. They contain the same neuron groups. They contain pyramidal cells, which are distinct with the apical dendrite, and inhibitory cells, which in this diagram are represented by these small round shapes. The layers are connected with each other. They also receive input from, this is the VPMPC, the taste thalamus. That's the basolateral nucleus of the amygdala. Those inputs come in specific layers, have preferences for certain cell groups. So we know this, and then we can go in and isolate, we can decide to look at the relationship between excitation and inhibition at the synaptic level by focusing on pyramidal cells and GABAergic cells. We use patch clamp recordings on them. We can record currents. some of the things that we know, during postnatal development, parvalbumin expressing inhibitory neurons begin to accumulate around them what are called perineuronal nets. What these are is molecules in the extracellular space that form a really tight network. And these have been discovered in many different cortical regions of the brain, and their formation is associated with limiting the ability of the brain to change in response to experiences. So once those have accumulated, the circuit is considered mature. Okay? The other thing that happens over the course of postnatal development is that synaptic transmission from inhibitory neurons increases in magnitude. So you have more inhibition and accumulation of these perineuronal nets that constrain the transmission and the connectivity of GABAergic cells. So what we decided to do was go into our system and figure out what happens during development and following the experience. So for this we use immunohistochemistry, and in this case, so this is a section of the brain, this is the gustatory cortex, this is somatosensory, this is olfactory cortex. The green cells that you see there are the GABA cells expressing parvalbumin, and the red accumulations that you see there are those extracellular matrix components that are clustering around the parvalbumin cells. So you see over there, there's a green cell with surrounded by red, it looks yellow. So those cells are mature parvalbumin cells, the others are still a little bit immature. This was an intermediate age. So when we count how many parvalbumin cells have been associated with perineuronal nets during development, what we see is that at postnatal day 17, before weaning, there's very few of them. And then at postnatal day 35 and 56, where we do the testing for behavior, there's a lot more. So the system has been developing, has been accumulating these perineuronal nets around parvalbumin cells. What we also see is that in the group of animals at P56 that had that 8 days exposure, the accumulation of perineuronal nets goes faster and is more. So there's more parvalbumin cells that have this accumulation at a comparable age, telling us that this experience is basically accelerating the maturation of the circuit. Other thing that we do is go see if this is also related to that increase in synaptic inhibition that I was telling you before. So if the transmission between GABAergic cells and excitatory cells increases, and so this we do with electrophysiology, with PET labs. So now we cut brain slices from animals that were naive to experience, an animal that had the 8 days of experience. We place microelectrodes in the cell body of these cells so that we have full electrical continuity of the membrane with our with our glass pipette and the electrode inside. We can measure currents now. We have a whole system which has amplifiers and other electronics that allow us to detect these currents. And then we use immunohistochemistry to make sure that the cells that we are recording are actually pyramidal cells that are glutamatergic. And so they have to be negative for GAD67, which is a marker for GABA. So the cells we are recording from are these red ones, they have no GABA. And what we want to measure is the inhibition on them. Okay, so how much inhibition they receive. And so here, this is what traces look like. These are inhibitory currents that are recorded from this, these peaks that go up and down. So in the naive animal, we have a certain number of them. In the animal that underwent that taste exposure, we have many, many, many more. And in that curve, it shows how many more. It looks small, but it's actually a pretty significant effect. What this is telling us is that the probability of GABA being released upon the arrival of an action potential is a lot higher in the animals that have been exposed to taste, which justifies why fewer pyramidal cells are active, because a lot of them receive more inhibition. So they are less able to fire action potentials. All right. So we have learned that with this experience, we have a change in the preference for sugar, we have a change in the decoding activity, changes in the activation of the circuit, fewer pyramidal cells are active, and this this reduced activation of pyramidal cells depends on a faster accumulation of perineuronal nets and an increase in strength, in magnitude of inhibitory synaptic drive. Now, what we wanted to know is that if this is real, right, and if it is limited to early development. So first question is, do we see that shift in the curve if we deliver the experience, if we expose the mice to that taste experience as adult instead of at the time of weaning, right? Or, you know, is the taste system plastic all the time, and so we just do the exposure later and we have the same. So what we did is we went back to our experience paradigm. Instead of starting it at postnatal day 22, we now started at postnatal day 27, when the mouse is a young adult. And so we keep the 5 weeks distance between the beginning of the experience and the testing. And so now we test them at postnatal day 90. Okay, and this is the usual just 8 days. So what we did see is that if we do this, we have no shift in the curve. So all of that I showed you so far depends on this experience coming early, when the animal is beginning to eat independently. Okay? So there is a sensitive window for the experience-dependent development of sucrose preference that depends, seems, on the maturation of the inhibitory circuit, the accumulation of perineuronal nets, the increase in strength that seems to constrain the system. So to validate that idea, what we thought is, what if we now make the inhibitory circuit immature in an adult animal? Would we be able to see the shifting curve again? Right? If you have a fully mature inhibitory circuit in a fully adult animal, and now you have a way, an experimental way, to go and bring those inhibitory circuits back to an immature state, would you now see an effect of experience comparable to what we saw in the early age? So this idea is based on some experiments that were done partly by me and partly by other groups in the visual cortex. I used to work in the visual cortex years ago when I was a postdoctoral scholar and at the beginning of my independent scientific life here at Stony Brook. And so there we knew that if you have a way to push the inhibitory circuit to be mature in adult, you can potentially reopen this sensitive window for visual cortical plasticity, which is very useful for kids that are born with misaligned eye, because it gives you time to correct it. So in taste is a little bit different. There's no work that was done in this system, and so we had to test it ourselves, right? So what we did here is we let the animal be weaned and grow. And then at postnatal day 44, we do a survival surgery. We implant a small, tiny little cannula positioned right on top of the taste cortex. And then what we do is at postnatal day 55, we inject an enzyme, the chondroitinase ABC, which is an enzyme that eats up the perineuronal nets and makes them disappear. So perineuronal nets are thought to be a marker of maturation. When they're not there, the idea is that the GABA cells become immature again because they're flexible enough, they can make more connection, they can make more synapses. So we were also testing that idea in the system. As a control, we have a group of animals that instead of receiving chondroitinase ABC, receives penicillinase, that pen there. Penicillinase is an enzyme, just like chondroitinase, but does not affect the perineuronal nets. So it's a neutral enzyme. It's just that it still has enzymatic activity, and we needed to control for that. Then what we do is we do the usual 8 days of experience, then back to the regular diet and testing at postnatal development. So this is a section of the brain of animals that have the cannula implanted. So these holes are telling you that our positioning is correct because the taste cortex is right here. And then we do immunohistochemistry and use these labeling methods to see the parvalbumin cells and the perineuronal nets. And you can see in the middle picture that all the red is gone in the chondroitinase-treated animals, whereas where there's penicillinase, we still have a lot of red. And those are just quantification bars that show you that the number of parvalbumin-containing GABA cells remains the same. It's just the perineuronal nets that go away. Okay? So now we have our exposure, and we go do our preference curve. And lo and behold, we see the shift. So it is really the accumulation of perineuronal nets around those parvalbumin cells that constrains the plasticity of the system and constrains the change in preference for sugar. So I'm coming down to my last slide, which is just a summary. What I showed you today is that the preference for sweet is sensitive to experience during a restricted sensitive window in postnatal development, and this restricted window starts at the time of weaning. We haven't really examined how granular this is and how far it is. I showed you that taste experience can change the activation of the gustatory cortex, of neurons in the gustatory cortex, and can change their ability to code for concentration of sugars, is actually making it better. And then what I showed you is that all of these changes in behavior and network activity depend on the maturation of inhibitory circuits in the gustatory cortex. If we can make them juvenile again in an adult animal, we can restore this shift in preference later in life. And so with that, I just want to thank my group, the people in my lab who did the work. In this particular work was done by Hilary Schiff, a postdoc in the lab. She is in the red shirt, so you can see her very well. And Maria Isaac, who's a student that recently joined my lab. She's not in the picture because she wasn't in the lab yet. And then the 2-photon imaging was done by Josh Kogan, right there on the side. He is a member of the lab run by Alfredo Fontanini, which is next door to ours, and we share the 2-photon system for doing experiments. And then those are my funding agencies, and then my former alumni and collaborators. And I'm happy to take any questions. Yes?
Is it possible that for the older mice, if you give it on a longer period of time, would the shift have definitely be a longer period of change in diet, or would it just stay the same?
So that's an excellent question, and it's something that we haven't really tested. It is possible. It's just that the problem is how much longer, right? It is a very interesting question. My intuition is that the taste system is plastic throughout life. In fact, we do change our preferences, we change our things, likes and dislikes, right, over the course of our lives. So the system is plastic. Whether this type of plasticity is the same across all ages, or you have a slightly different type that comes in once all of your preferences are set and now you modify them, is unknown. Whether you need, say, 3 weeks of exposure instead of 8 days is unknown. This is really the first and only piece of work on something like this. Yes?
Do you incorporate things like change in taste to physiological changes in the body, like the phenomenon of women who are pregnant having certain cravings for things, or menopause causing changes in things. Does that figure into any of your work?
Not yet.
Oh, okay.
So there's a few things that we're trying to do. We have, a whole line of research. We're looking into signaling molecules that signal satiety, for example, because your satiety depends very much on not only how much you ate, but how much you ate of that specific food. Sometimes you satiate with sugar, but if they offer you something salty, you're still hungry, right? So how that pans in, there's some ideas in the hypothalamus that have to do with metabolism, but there's also sensory aspects which have to do with the taste cortex. The effect of hormones on taste, I have never looked into that. I don't think there's many people that look into that because it's complicated. You have to track the cycle of the mice. You can do it, it's not impossible, but it adds complications and tracking exactly what the levels are across animals. So these are all interesting questions, and ultimately this is where, dietary preferences based on experiences where my lab is taken off. It's just that it takes a lot of people and a lot of funding to expand it in all the directions we want to go. You can ask me questions even later if you think about them. Just shoot me. I'll reply.
Another question for Maffei. All right. Then thank you so much.
Thanks.